Hi students, welcome to Smart Locus. In this video, we will see the chapter Transport in Plants. In the introduction, mainly two types of transport, that is small distance transport and long distance transport. Description is given. Small distance transport is mainly by diffusion, that is passive, then cytoplasmic streaming, then active transport. Long distance transport is known as translocation, and it occurs through mass flow or bulk flow. Then means of transport. Means of transport includes diffusion, that is simple diffusion, then facilitated diffusion, then active transport. Here, simple diffusion and facilitated diffusion are passive processes, doesn't require energy. Whereas active transport is an active process, that means it requires energy, ATP. Then, comparison, first point. Diffusion and facilitated diffusion are downhill transport mechanisms. That means here the movement takes place from higher concentration to lower concentration along a concentration gradient. Whereas in active transport, it is uphill. That is from lower concentration to higher concentration. Then, diffusion, facilitated diffusion, if we compare, one difference is hydrophobic molecules, that is plasma membrane allows simple diffusion of hydrophobic molecules, whereas hydrophilic molecules find it very difficult to cross the membrane. Their movement has to be facilitated with the help of proteins present in the membrane. That is known as facilitated diffusion. And if we compare facilitated diffusion and active transport, few similarities are, in both the cases proteins are present. Proteins present in plasma membrane are involved in both facilitated diffusion and active transport. And the protein involved in facilitated diffusion known as porin, whereas in active transport it is pump. Then both are highly selective. Why? Because proteins are specific and both are sensitive to inhibitors that attach on or that bind on protein side chains. And in both the cases, transport saturates when all the proteins are used up. And both are under hormonal control. Then few specific points in diffusion. It is the only means of gaseous transport in plants. And diffusion, it is not dependent on a living system. It takes place in living systems. And the various factors that influence the rate of diffusion are concentration gradient, permeability of the membrane, pressure, temperature, solubility in lipids, then size of the diffusing particle. Then a few specific points in facilitated diffusion. Channels formed by the membrane proteins are of two types. It can be either permanently open or controlled, that is gated. And then membrane protein involved in facilitated diffusion. Example, porins. Porins are present in the outer membrane of mitochondria, chloroplast, and in the cell membrane of bacteria. Water channels are formed by 8 different types of aquaporins that facilitate the movement of water. Types of facilitated diffusion. Some transport proteins facilitate or allows diffusion only when two types of transport molecules are present. In this case, if the two types of molecules moves in the same direction, then it is known as import, and if they move in the opposite direction, then it is known as antiport. So in both the cases, see here, two types of molecule, for example, A and B, C and D. The movement of A is dependent on movement of B. Similarly, movement of transport of C is also dependent on transport of D. Whereas in uniport, one molecule moves across the membrane independent of the other type of molecule. So here, transport of P is not dependent on transport of Q. Next, about plant-water relations. In case of plant cells, 85 to 90% of the volume, see in herbaceous plants, only 10 to 15% forms of the fresh weight forms dry matter. That means 85 to 90% forms water only. Without the presence of water, plant physiology is not possible, plant life is not possible. Because water is required for all the activities. Excess water in plant cells is known as turgidity, which is necessary to maintain the normal shape and structure of plant parts. Then, in case of watermelon, 92% water only. A mature corn plant absorbs 3 liters of water per day. A mature mustard plant absorbs water equal to its own weight in about 5 hours. So see, because of this huge requirement, water often forms a limiting factor for plant growth. Next, about water potential. Water potential is a concept fundamental to understanding water movement. The two important determinants or components of water potential are solute potential and pressure potential. Water potential is denoted by the Greek symbol psi. Psi W, water potential; psi S, solute potential; psi P, pressure potential. Is nothing but kinetic energy of water molecules. And water moves from a region of higher potential to a region of lower potential. Then, pure water has the greatest water potential. Water molecule has maximum kinetic energy in pure water. Then, water potential is expressed in pressure unit, that is Pascal. Then, by convention, water potential of pure water at standard temperature and pressure is taken to be 0. Solute potential, that is osmotic potential, psi S. Solution. Water potential of solutions are always less than 0. The magnitude of this lowering due to the dissolution of solute molecules is known as solute potential. It is always negative. The more the solute molecules, the lower is the psi S, that is more negative is the psi S. For a solution at atmospheric pressure, psi W equals psi S. Then next, about pressure potential, that is psi P. Pressure that develops in plant cell due to the entry of water, it is usually positive. But negative pressure potential plays an important role in the upward conduction of water and minerals through xylem by transpiration pull. If we consider psi W of a plant cell, psi W means water potential of a plant cell equals psi S plus psi P. These are the two determinants. Next, about osmosis. Is nothing but diffusion of water molecules through a semi-permeable membrane. And under this title, the first point given in, the two important points given in NCERT are, the first point is, vacuolar sap contributes to osmotic potential or solute potential of plant cell. Plant cell has large central vacuole. Second point is, cell membrane and tonoplast. Tonoplast is the membrane of vacuole. These two are the two determinants of the movement of water, both towards the inside and towards the outside. Then, the rate and direction of osmosis depends on both pressure gradient and concentration gradient. It's about the Sil Funnel Experiment. A sucrose solution taken in a Sil funnel is separated from water taken in a beaker by egg membrane. Here, water moves from a region of higher concentration to lower concentration, or higher potential to lower potential. That means water enters from the beaker to the Sil funnel. That is, the observation would be the water level inside the Sil funnel rises. This can be prevented by applying pressure inside the water column in the Sil funnel. That pressure is known as osmotic pressure. That is, the pressure which prevents water molecules from diffusing. This is actually a function of the solute. That is, the more the solute concentration, the more higher pressure is to be required to prevent the entry of water molecule. That is, here the osmotic pressure and osmotic potential are equal in the sense numerically equal One is positive, the other is negative. Next, about plasmolysis. The behavior of plant cell with regard to water movement depends on the external solution. If the external solution balances the osmotic pressure of the cytoplasm, then that solution is isotonic. If the external solution is more concentrated, then it is hypertonic. If the external solution is more dilute, then it is hypotonic. Let us see what would be the behavior of plant cell when it is placed in the three different types of solutions. First, in hypertonic solution. Hypertonic solution, if we place plant cell, first exosmosis takes place. Water moves out. The protoplast shrinks because plasma membrane gets detached from the cell wall. Protoplasm shrinks. This is known as plasmolysis. So, in hypertonic solution, plant cell gets plasmolysed. And there is a question in NCERT. What occupies the space between shrunken protoplast and cell wall in a plasmolysed cell? The answer is external solution, because cell wall is freely permeable. Then second, behavior in isotonic solution. When we place plant cell in isotonic solution, both endosmosis and exosmosis takes place at equal rate. That means no net movement of water takes place. Plant cell loses turgidity, there is no excess water in plant cell. And this condition is said to be flaccid. So, plant cell becomes flaccid when it is placed in isotonic solution. There is a question in NCERT. What would be the Psi-P, pressure potential of flaccid cell? 0. It won't be able to exert pressure on the cell wall. Third, hypotonic. Plant cell in hypotonic solution. Endosmosis occurs. And this plant cell becomes turgid. Turgidity exerts pressure on the cell wall. That is known as turgor pressure. And here, among the three conditions, plasmolysed, flaccid and turgid, turgidity is necessary for the normal functioning, normal physiological activity of plant cells. Imbibition. Imbibition is a special type of diffusion. Movement takes place along a concentration gradient. And here absorption of water molecules by solids or colloids, causing them to increase in volume. A typical example is swelling of dry seed or wood when it is placed in water. This process is necessary for seed germination. Then, next about long distance transport. It takes approximately 2.5 seconds to move water molecules through a typical plant cell. But large and complex organisms, in case of large and complex organisms, water has to be moved for a longer distance. That requires mass flow or bulk flow. Mass flow or bulk flow, the main difference between mass flow and diffusion is that in mass flow, the movement is based on pressure gradient, whereas in diffusion, a movement is based on concentration gradient. And here, all the molecules moves at same pace. And the movement of particles through or transport of particles through mass flow is known as translocation. This mass flow can be achieved either through a positive hydrostatic pressure gradient, that is, example, a garden hose, or through a negative hydrostatic pressure gradient, example, suction through a straw. Then, xylem, phloem. Xylem conducts mainly water, minerals, then some organic nitrogen and hormones. These are transported through xylem. Similarly, phloem sap. Phloem, both organic and inorganic solutes are there. Phloem sap mainly consists of water and sucrose. Mainly. Then, along with this, other sugars are there, amino acids are there, then hormones are also there. Next, about absorption of water. Root hairs are responsible for absorption of water. And water absorption is purely by diffusion. After absorption, further movement of water in root into the deeper layers takes place by two pathways, that is apoplast pathway and symplast pathway. Apoplast means a system of adjacent cell walls present throughout the plant cell except in the Casparian strips of root endodermis. In apoplast pathway, the movement takes place through intercellular space and cell wall, and apoplast movement does not involve crossing the cell membrane. And since the movement takes place through intercellular space and cell wall is freely permeable, it does not provide any barrier to water movement. As a result, water movement is through mass flow of water takes place. Symplast means system of interconnected protoplast, and here water movement takes place through plasmodesmata, the open channel or cytoplasmic connections between plant cells, and here the movement involves crossing the cell membrane, and it is aided by symplastic movement is aided by cytoplasmic streaming. Since it has to cross the cell membrane is semi-permeable only, the movement is comparatively slower. Most of the water flow in roots takes place through apoplast because the cells of the cortex are loosely packed. When water reaches the root endodermis, it cannot penetrate the Casparian strips, the waxy coating. Then water will be directed to cell wall regions that are not suberized. Then water enters into the cytoplasm of endodermal cell by crossing the cell membrane, and further movement occurs through symplast. Then again it has to cross the cell membrane to finally reach the xylem, that is the vascular tissue. So we can conclude that water movement at endodermis is ultimately symplastic. In young developing roots, water enters directly into xylem vessels and tracheids. Vessels and tracheids are non-living. That means apoplast. The next about mycorrhiza. Mycorrhiza is nothing but symbiotic association between fungus and roots of higher plants. And here fungal hyphae increases the surface area of absorption. In return, plants provide nutrition. Some plants cannot germinate. For example, pinus cannot germinate and establish in the absence of mycorrhiza. The next point is about upward conduction of water and minerals. That is water movement up. Here under this title, the first point is root pressure. As various mineral ions are actively absorbed into root, that means the vascular tissues, water follows by passive means, that is diffusion. Then pressure develops in root or pressure increases in root. This positive pressure is known as root pressure, gattation. That gattation takes place mainly at night and early morning. That is, water molecule oozes out from small openings, that is specialized openings known as hydathode, present at vein endings of leaves in herbaceous plants. And this gattation is actually due to root pressure. Then exudation, that is nothing but dose of water droplets from injured part. That is also due to root pressure. So the greatest contribution of root pressure in plant physiology is to re-establish. It helps to re-establish the continuous water column in xylem, which often breaks under transpiration pull. Transpiration pull, that is the driving force for the upward conduction of water and minerals. That is, according to cohesion, tension and transpiration pull model, transpiration pull is responsible for the upward conduction of water and minerals. Then next about transpiration. Less than 1% of the water that reaches the leaves is used for photosynthesis and plant growth. That means most of the water is lost through transpiration. And this Transpiration is nothing but loss of water in the form of water vapor, and it takes place mainly through stomata present in leaves. And stomata, the small opening, stomata are involved in gaseous exchange also, the mechanism of opening and closing of stomata. Usually in plants, stomata opens during daytime and closes during night, except in plants with the desert adaptations. In desert plants, just opposite. And stomata opens when the guard cell becomes turgid. When the guard cell becomes turgid, the outer thin walls of the guard cell bulges out, and it forces the inner thick elastic wall into a crescent shape with the help of radially arranged cellulose microfibrils. That leads into opening of stomata. The next about closing of stomata. When the guard cell becomes flaccid, the inner wall regains its original shape, which leads into the closing of stomata. Next about factors that influence the rate of transpiration. External or environmental factors are light, temperature, wind speed, humidity. Internal or plant factors are number of stomata, distribution of stomata, monocot, dicot, distribution differs. Then percent of stomata open. Then water status of the plant, whether the plant is having excess water or not. Then canopy structure, whether the leaves, canopy means the branches. Leaves are compactly packed or loosely arranged. Transpiration driven ascent of sap, that is upward conduction, is mainly due to three physical properties of water, that is cohesion, adhesion and surface tension. Cohesion, attraction between water molecule. Adhesion here, attraction of water molecules by polar surface, that is tracheids and vessels. Then surface tension, water has more surface tension in its liquid phase. These three physical properties gives high tensile strength and capillarity to water molecule. Tensile strength and capillarity to water molecule. Tensile strength here, tensile strength is nothing but the ability to resist a pulling force. Here the pulling force is transpiration pull. This helps to maintain a continuous water column in xylem without breaking. And capillarity, ability to rise in a capillary tube. It is aided by the smaller diameter of the tracheary elements, that is xylem vessels and xylem tracheids. The next point, transpiration and photosynthesis, a compromise. Which are the advantages of transpiration? Transpiration pull is necessary for the absorption and transport of water and minerals. That is the driving force. Then it supplies water for photosynthesis. Then it cools leaf surfaces. In some plants 10 to 15 degree. Then it helps to maintain the shape and structure of plant parts by keeping the cell turgid. Then the evolution, the main objective of the evolution of C4 plant is to maximize the photosynthesis, that is carbon fixing efficiency, and to minimize water loss. They are, that is C4 plants are twice as efficient as a C3 plant in terms of making sugar, and they loses only half as much water as that of a C3 plant for the same amount of carbon fixed. Uptake and utilization of mineral ions. Minerals. All minerals cannot be passively absorbed. That means active transport is also required because of two reasons. The two reasons are: one, minerals are present in soil as charged particles, that means ionic form. So the movement of these ions require energy, that is active transport. Second point, concentration of mineral is more in plant cell, and the requirement is there in plant cell. That means mineral ion have to be moved from lower concentration to higher concentration, that is uphill transport. These are the two reasons why absorption of minerals involve active transport also. Then, next point, control point. Control points are nothing but proteins present in the cell membrane of endodermal cell. Actually, this protein adjust the type of solutes to be absorbed and quantity of the solutes to be absorbed. So both are decided by a protein present in cell membrane of endodermal cells. These proteins are known as, or they act as, control points. Next point, translocation of mineral ion takes place along with the transpiration stream by transpiration pull. And the chief sink means the place where it is utilized or required. The chief sinks of mineral ions are nothing but growing regions, which includes meristems, then developing fruits, seeds, storage organs, then developing flowers. Next, about phloem transport. Phloem transport is either bidirectional or multidirectional. And phloem sap consists mainly of water and sucrose. Along with this, other form of sugars, amino acids and hormones can also be seen. Mass flow hypothesis or pressure flow hypothesis. Here first, phloem loading takes place at the source, that is at the mesophyll. At mesophyll, the immediate product of photosynthesis is glucose, which will be transported in the form of sucrose. So from mesophyll, at the source, sucrose enters into companion cell, then into sieve tube. So mesophyll, companion cell, sieve tube. This is known as phloem loading, and this process requires energy, that is active transport. So phloem loading is by active transport. And this phloem loading creates a hypertonic condition in the phloem. As a result, water, by osmosis, water enters into the phloem from adjacent xylem, which creates osmotic pressure. That means a pressure gradient will be created between source and sink. As a result, mass flow of nutrients takes place from source to sink through sieve tube. Then, at the sink means the place where the food material is utilized or stored. Phloem unloading takes place. That is, sucrose moves out of the phloem into the cells where it is utilized. And this phloem loading also requires energy. That is, it is also by active transport. The next about Gatling Experiment. The main objective of this experiment is to prove that food materials are transported through phloem. Here, the experiment involves removal of a ring of bark from tree trunk. The observation after few days would be the point of girdling, above the point of girdling becomes swollen. That is due to the deposition of food materials. That is, the downward transport of food materials to root is blocked because phloem is lost.
If you are not yet subscribed, please subscribe by pressing the subscribe button below and press the bell button also to get all the notifications from the channel. If you think this video is really useful for NEET preparing students, please share it. Thanks for watching.